We have all been taught the law of muscle growth. It goes like this: you are born with a set number of muscle fibers. When you lift weights, those fibers get thicker, like inflating a balloon. When you stop, they shrink. But you never, ever get more fibers. Welcome to Dr. Levitas. This is called hypertrophy. And it is the accepted science. But what if that law is wrong? What if there is a secret biological backdoor that allows you to not just make your fibers bigger, but to actually split them in half and create entirely new ones? Today, we are investigating the holy grail of bodybuilding, hyperplasia. We are going to dive into the controversial science of fiber splitting, look at the extreme animal experiments that produced 300% growth, and ask the big question. Do massive bodybuilders simply have bigger cells than you, or have they mutated to have more of them? Also, please take a second to like the video and leave a comment. Your engagement is a huge help to the channel. First, let's define the terms and conditions. Hypertrophy is what happens to 99% of people in the gym. You stress the muscle, you eat protein, and the individual muscle cell adds more contractile units, more sarcomeres, and fluid, more sarcoplasm. In short, it swells. But eventually, a cell hits a genetic ceiling. It can only get so big before it becomes inefficient to sustain. Hyperplasia is different. It is the increase in the number of cells. Skin cells do this constantly, like when they have to heal a cut. Liver cells do this. But muscle cells are post-meiotic, meaning they supposedly lost the ability to divide after you were born. If hyperplasia is real in humans, it effectively removes the genetic ceiling. Instead of trying to overinflate a single balloon, you are essentially adding more balloons to the pack. Why do we think this is possible? Because of some birds. In the 1990s, researchers like Dr. Jose Antonio conducted a series of extreme experiments. They took birds, quails, and attached a weight to one wing that stretched the muscle continuously for weeks. We aren't talking about a 1-hour workout. We are talking about a 24/7 loaded stretching. The results were biologically impossible by standard models. The stretched muscle didn't just grow a little. It grew by 318%. When they put the tissue under a microscope, they realized that hypertrophy, the cell size, couldn't account for all that mass. They counted the fibers. The stretched wing had significantly more fibers, and in some cases up to 82% more. The extreme, chronic tension had forced the muscle to split its fibers to survive. It was an adaptation to extreme stress. So, does this happen in humans? This is where it gets a little messy. You can't ethically stretch a human's arm for 30 days straight, and you can't slice open a pro bodybuilder's bicep to count the fibers while he's alive. However, we have some clues. Some studies comparing elite bodybuilders to powerlifters and control subjects have found something interesting. The bodybuilders' individual muscle fibers weren't actually that much bigger than the powerlifters. In fact, some were smaller. But the bodybuilder's total muscle size was massive. If their cells aren't bigger, but the muscle is huge, the only mathematical explanation is that they have more cells. This suggests that years of high-volume, high-tension training, and let's be honest, likely weird compound use, may trigger hyperplasia in elite humans. How does a muscle cell replicate? It doesn't go through mitosis like a skin cell. It undergoes fiber splitting. The theory is that when a fiber reaches a critical size threshold, when it becomes too big for the oxygen and nutrients to diffuse into the center, it becomes metabolically unstable. To save itself, the fiber develops a fissure down the middle. It splits into two smaller, more efficient fibers. Now, you have two fibers that can both grow back to the original size. This effectively doubles your growth potential. But reaching that critical threshold of size requires a level of intensity and volume that most natural trainees simply never reach. We cannot talk about hyperplasia without talking about hormones. IGF-1, or insulin-like growth factor 1, is the primary driver of this process. When you train, your liver and muscles produce IGF-1. IGF-1 activates satellite cells. These are the stem cells of your muscle. Usually, they just fuse to existing fibers to make them bigger. But in the presence of extremely high levels of IGF-1 and growth hormone, there is evidence that satellite cells can fuse with each other to form entirely new fibers. We call them myotubes. This is why professional bodybuilders, who often use exogenous growth hormone and insulin, display that grainy, dense look that seems different from natural muscle. They may be chemically unlocking hyperplasia. Is there a natural way to mimic this? Maybe there is. It comes back to the pump. For years, we thought the pump was just cosmetic. But research suggests that cell swelling is an independent growth signal. When a cell is engorged with fluid, with the sarcoplasm, the internal pressure threatens to burst the cell membrane. The cell perceives this as a threat. To reinforce the structure, it increases protein synthesis and, theoretically, may trigger splitting to reduce the internal pressure. This validates high representative training. The burn and the pump aren't just for show. They are creating the hydrostatic pressure needed to threaten the cell into dividing. If we want to replicate the bird study, but without the torture, the closest technique we have is intraset stretching. This involves taking a muscle to failure, and then immediately, while it is pumped full of blood, holding it in a fully stretched position with weight. Here is a clear example. Do a set of dumbbell flies to failure. Then, let the weights hang in the deep stretch position for 30 to 45 seconds. This combines the cell swelling, the known pump, with the mechanical tension, the stretch. You are trapping blood in the muscle while pulling the fibers apart. It creates a hypoxic, high-tension environment that mimics the conditions seen in the animal hyperplasia models. Here is the doctor's verdict, pushing the biological limit. Is hyperplasia a guarantee? No, the science is still debating the extent to which it occurs in average humans. But the evidence is strong enough to suggest that if you want to break past your genetic limit, you can't just lift heavy. You have to lift differently. You need to chase the pump, you need to endure the extreme stretch, and you need to push your muscles to a place where they have no choice but to split to survive. Let's grow beyond the limits. I'm Dr. Levitas, helping you mutate your potential. Watch this video suggestion, it was especially picked for you. Also share, comment, give it a thumbs up, subscribe, and hit the bell icon. It helps a ton with the algorithm. Remain tuned for future videos to empower your health and fitness journey. So, stay healthy, stay happy, and keep going.